Can I have somebody confirm you can hear me?
I can hear you.
Thank you. See. Should be on chapter 6, but I think we're on chapter 5. All right, before we begin, are there any questions? Not hearing anything, so let's begin. Let me share my screen. I'm sure we're not there. Have to be further than that. Where do I have two here? There we go. That looks right. About 565. All right. Let me share my screen here. All right, should be seeing my screen now. So today is August 5th, and we should be on chapter 6. We're not there yet. We have to finish chapter 5. There is a quiz this Wednesday, quiz 3, and it'll be over lab 5 and 6, chapter 4, and the end of chapter 3. Any questions about any of that? I'm going to switch my glasses there. I wasn't able to see with those glasses. They were a little weaker in power and much older. There is a lab today. We're going to talk about lab 7. Hopefully later tonight, probably pretty late, sometime after the lab, probably a little after 10, so maybe even after midnight. I'll send out the information about the unknown project, including what your unknown number is and your first 3 unknown results. Any question about any of this? All right, let's finish chapter 5 and begin chapter 6. I need to get quiz 2 graded. I don't think I've looked at quiz 2 at all. But I did get some grading done. I did get the lecture assignment graded and then the lab for last week graded. So I have that we were talking about lipid and protein catabolism, and I mentioned that a problem with microbes as well as your cells in digesting lipids and proteins is these large molecules cannot get across the cell membrane. And so cells solve this problem by secreting enzymes extracellularly to digest the large molecules, break them into the monomers, and then the monomers can come across the cell membrane. Any question about any of that? And for your cells, the monomers, as well as bacteria, the monomers come through transport proteins. Meaning even small amino acids cannot get through the cell membrane. They need to get a transport protein to bring them across. But a large protein can't get through the cell membrane. So microbes, as well as your cells, produce lipases and proteases. They break down lipids and proteins to monomers. Lipids are broken down into glycerol and the fatty acids. The glycerol is catabolized by glycolysis. The fatty acids are catabolized into 2 carbon chunks, which then enter the Krebs cycle. Amino acids are brought across the cell membrane, and then the amino group NH2 is broken off, has to be removed, and then the remaining part is converted mostly to sugars, which then enter the Krebs cycle. I think I've got a picture of that in the next slide. Let me blow this out. So lipids broken down into glycerol, fatty acids broken down into two carbon chunks, converted into acetyl coenzyme A, which then enter the Krebs cycle. The glycerol comes midway through glycolysis. And amino acids, depending on which amino acid they are, they can be the amine group is removed, and then the amino acid, depending on the amino acid, can come in the end of glycolysis, converted into pyruvic acid. Some amino acids are converted into acetyl coenzyme A, and some are converted into a later product in the Krebs cycle. The point is that they all enter either glycolysis or the Krebs cycle, and that way carbohydrates, lipids, and proteins can be digested. I didn't say it, but carbohydrates, I think I've said it before, all carbohydrates are essentially broken down into glucose. And if they're another sugar, that sugar will be converted into glucose. Very few sugars enter glycolysis at another point other than glucose. I don't remember which one which one is not converted into glucose, but there is one sugar which enters glycolysis a little bit after glucose. It might be fructose, but I'm not sure. No, it's not fructose. Fructose is converted by the liver into glucose. I'm not sure about bacteria, but in humans, fructose will be converted into sugar, glucose, but in the liver. All right, the complete metabolic oxidation of glucose in aerobic respiration is the most efficient way to harness the energy from glucose and make ATP. The ATP is used for various cellular processes, including active transport across the cell membrane, movement of the cell, or like the flagella of the cell, or for humans, the movement of the muscle cell, as well as energy for the synthesis of new cellular components, meaning anabolism, making a larger molecule. Most cellular ATP is actually used for the latter, anabolism, and includes the biosynthesis of proteins, carbohydrates, and lipids, as well as nucleic acids, the four molecules of life. Any question about any of that? All right, I've talked about anabolic and catabolic pathways as separate and independent pathways, but in reality, they are joined by common intermediates. An anabolic pathway is a metabolic pathway that can function in both directions. They bridge the pathways involved in carbohydrate, lipid, protein, and nucleic acid metabolism. An amphibolic pathway is one that runs in both directions, such as we mentioned that a pyruvic acid can be converted into acetyl coenzyme A. Well, acetyl coenzyme A can go reverse and be converted into pyruvic acid. In fact, this amino acid, if you're a couch potato, the body doesn't like burning proteins and amino acids. So if you're a couch potato, that protein will be broken down into amino acid. And then if the body has time, that amino acid will be converted into oxaloacetic acid. And instead of going in the normal Krebs cycle this way, burning the, where's the previous? Burning the oxaloacetic acid, the oxaloacetic acid can be converted into this one, this one, this one, going reverse in the Krebs cycle, converted into acetyl coenzyme A, which I'm not sure why it would go that way when actually, well, Yeah, we don't want to make, well, I don't know why it can't go that way to make the acetylcoenzyme A. I suppose it could, but it could go this way as well. And then the acetylcoenzyme A can be converted into pyruvic acid, converted into this way, converted into that way, converted in that way. You see, these are uh, triangular points. So this reaction can go this way, but it can also go this way. It can also go that way. We won't talk about that. And let me see if I can get this out of the way now. And then it converted into that, converted into that, converted into glucose. And then the body will send glucose down aerobic respiration in the normal way. So that is how most couch potatoes burn proteins as well as lipids. Will be converted into some, well, fatty acid converted into acetylcoenzyme A, then go this way up to make glucose. And then glucose will be sent down aerobic respiration in the normal way. It is true that a marathon runner can burn lipids directly in their marathon run. However, the marathon runner does not burn lipids until 45 minutes into their run. So even at the start of the marathon run, even the marathon runner will convert the lipids into glucose and then send glucose down in aerobic respiration. And it's true the marathon runner is also using carbohydrates and glucose initially in the run in aerobic respiration. But when that runs out, they can burn lipids directly, but they don't do that until 45 minutes into their run, in which case the lipid will be converted into glycerol and fatty acids, and then the fatty acids will enter converted into acetylcoenzyme A, enter the Krebs cycle to be burned directly. Any question about that? And I'm sure a marathon runner can burn proteins directly too, but I don't know when in their run that happens. So initially, the proteins will be converted into glucose, and then glucose sent down the aerobic respiration pathway in the normal way. But sometime into the marathon run, they will burn the proteins directly, and then the Krebs cycle will happen in the normal way with an amino acid. Any question about any of that? All right, so a lot of the pathways can go both ways, and we call them amphibolic pathways, and that bridges the metabolism of carbohydrates, proteins, and fatty acids. And you actually already know this. If you eat too much carbohydrate, your body will initially store it down as glycolysis in your liver, but once your liver becomes full of glycolysis, what does your body store that carbohydrate as? Come on, somebody's got to know because everybody knows this. You're not going to store it as carbohydrate. What are you going to store it as? Nobody's going to suggest it, huh? Well, I'll mention with protein, if you eat a high protein diet and you have too much protein, it will be converted not into, it will initially be converted into glucose and then stored down as glycolysis. But if you eat too much protein, you will actually convert that protein into lipid, and then you'll store that lipid as body fat. So you already know that proteins, carbohydrates, and lipids are all interconnected. And this is how they're connected. The protein will be broken down to amino acids, and then the amino acids will initially be converted into glucose. But after that, the amino acids will be converted into fatty acids and glycerol, and then stored as lipids. All right, let's briefly talk about photosynthesis. I've got 2 slides to talk about photosynthesis. Photosynthesis is the conversion of light energy to chemical energy. chemical energy may be used to convert atmospheric CO2 to glucose. That we call carbon fixation. But photosynthesis does not always make glucose. And photosynthesis is performed by obviously green plants. Everybody knows that, by algae, but also some photosynthetic bacteria, including cyanobacteria. Photosynthesis is a mechanism for synthesizing organic compounds. Without photosynthesis, animals and microbes would not have something to eat, meaning all living life on the planet, with the exception of maybe one ecosystem on the planet, requires photosynthesis to provide the majority of the food or the glucose in the, what do you call that? The ecosystem, in the niche in the ecosystem. There is one niche on the planet that does not rely on photosynthesis. It is the deep sea vents. And what is actually making the most food in the deep sea vents, meaning the majority of the food, is by chemosynthetic bacteria engaging in chemosynthesis, converting an inorganic compound, pulling electrons from an inorganic compound, and then using those electrons to make glucose. But other than the deep sea vents, all ecosystems on the planet Earth primarily rely on photosynthesis to make glucose for animals and microbes to feed on, as well as obviously the photosynthetic organisms. Any question about any of that? And if you say, well, I don't rely on photosynthesis because I eat cows, meaning beef, well, where do you think the cows get their food? From eating the grass, which is obtained from photosynthesis. Photosynthesis only occurs in photosynthetic organisms. These cells contain a light-trapping pigment, collecting the light and then changing it into a chemical energy form, such as ATP. One of these photosynthetic pigments you actually already know, and that is chlorophyll. When light strikes the electrons in chlorophyll, it causes the electrons to move out of chlorophyll. And then that light energy can be converted to chemical energy by an electron transport chain. And ATP and NADPH are generated. NADPH is similar to NADH. It's just that in photosynthetic organisms, they have NADPH instead of NADH. So these molecules are made in photosynthesis, but oftentimes ATP and NADPH are used up to synthesize glucose from CO2 and water. And this reaction we call carbon fixation, where you fix the CO2 to make glucose. However, photosynthesis does not always make glucose. There are two types of photosynthetic reactions, the cyclic photosynthesis and the non-cyclic photosynthesis. You already are familiar with the non-cyclic photosynthesis reaction, making oxygen, and you do need to know this one, where 6 carbon dioxide molecules get together with 12 water molecules in the presence of light to make glucose, C6H12O6, 6 oxygen molecules, and 6 water molecules. Sometimes you'll see this reaction where they've subtracted the 6 water molecules here and then subtract the 6 water molecules there. I prefer this reaction though, because if we look at where all the 12 atoms of oxygen gas come from, they all come from water. So all of this oxygen comes from the oxygen and water. So you need 12 water molecules into the reaction to get the 6 oxygen, and then water has to come here, meaning none of this oxygen comes from the conversion of CO2. All the CO2 is converted into glucose. And then the water is converted into oxygen. And I suppose the CO2 and the water also makes water over here. You do not know the non-cyclic photosynthesis reaction, which does not make oxygen, and that's 6 CO2 plus 12 H2S, hydrogen sulfide gas, in the presence of light, can make glucose, as well as 12 sulfur atoms, and then 6 water molecules. You don't need to know this, I won't quiz you on it, but there is another photosynthesis reaction that does not make oxygen. And then there's another photosynthesis reaction that does not make glucose. In this reaction, ADP, an inorganic phosphate, in the presence of light, creates ATP. All photosynthetic cells can engage in either cyclic photosynthesis or non-cyclic photosynthesis. Like green plants, whenever they need glucose, they'll engage in non-cyclic photosynthesis. But if a green plant needs ATP, it will engage in cyclic photosynthesis. Any question about any of that? Let's talk a little bit more about cyclic photophosphorylation, meaning cyclic photosynthesis. Light excites the 2 electrons from chlorophyll, which then move out of the chlorophyll. The chlorophyll is in the proteins of photosystem 1 here, why it's kind of green. The electrons then move in this protein in a membrane, which then the electrons move to this protein in the membrane. Then the electrons are moving through this protein in the membrane. They give the protein the energy it needs to pump hydrogen ions from this side of the membrane to that side of the membrane. The electrons then move from this protein to that protein in the membrane, and then the electrons move to this protein in the membrane. And then the electrons flow back to chlorophyll, the molecule that they initially came out of. And that's why it's called cyclic photophosphorylation, because the electrons move in a cycle, returning to the place from where they originated. Any questions about any of that? Well, this should look a little bit familiar to you because it's very similar to the electron transport chain of aerobic respiration. I'm not seeing the slide here. There we go. It's similar to the electrons moving to this protein in a membrane, to that protein, to that protein, to this protein, to this protein, only it's shorter, and only one protein of cyclic photosynthesis can pump the hydrogen ions from this side of the membrane to that side. In aerobic respiration, we have 3 proteins doing it. But the point is, the electrons flow through proteins in a membrane, and when they're flowing, they cause hydrogen ions to move from one side of the membrane to another. And that is how the ATP gets generated both in aerobic respiration and in cyclic photosynthesis. These hydrogen ions create a gradient on this side of the membrane. Then they want to flow from this side of the membrane to, oops, sorry, got it on the wrong side here, from this side of the membrane to that side of the membrane. And the only place the hydrogen ions can do that is through this channel protein. And when the hydrogen ions flow through this protein, they give the protein the energy this protein needs to make ATP from ADP, an inorganic phosphate. Any question about any of that? And that's the same as in aerobic respiration. But the hydrogen ions are pumped from this side of the membrane to that side of the membrane, and then the twofold gradient of hydrogen and positive charges want to move to this side of the membrane so that potential energy is converted into the energy of ATP when the hydrogen ions move through this protein. In the prokaryote cell and aerobic respiration, this is the cell membrane, and this out here is outside the cell membrane. So in most prokaryote cells, this is the cell wall. And that is true for both aerobic respiration or cyclic photosynthesis. This is the cell wall where the hydrogen ions are pumped into, and then they come through this protein to make ATP. Now in eukaryotes, this doesn't happen in the membrane of the cell membrane. It happens in the inner membrane of the chloroplast. So cyclic photophosphorylation happens in the inner membrane of the chloroplast. And that's similar to eukaryotes with aerobic respiration, where it happens in the inner membrane of the mitochondria. Any question about any of that? All right, so let's have the last slide of chapter 6, requirements of ATP production. The electron's donor can vary. It can be glucose, like it normally is in aerobic respiration for our cells, but in chemosynthetic organisms, the electron comes from an element other than oxygen, like sulfur, ammonia, hydrogen gas. The electrons are stripped from that chemical, and then the electrons are carried by an electron carrier molecule, and the electrons are stripped from glucose also in aerobic respiration, carried by an electron carrier molecule, which are NAD plus or FAD in aerobic respiration. But the electron donor for photosynthesis comes from when light hits the photosynthetic pigment, the electrons in the pigment begin moving. And the electron carrier is a little different. It's NADP plus instead of NAD plus. Along the way, ATP is generated. And then the electron acceptor, the final molecule to accept the electrons, can also vary. For aerobic respiration, the final electron acceptor is oxygen. For anaerobic respiration, the final electron acceptor is a molecule other than oxygen. It depends on the form of anaerobic respiration. It could be nitrite, could be nitrate, it could be sulfate, it could be iron. And then for fermentation, it's going to be an organic compound like pyruvate, or what pyruvate converted into. And since it's the pyruvate which accepts the electrons, it'll be the pyruvate. And it's not listed here. I don't know why, because they've got photosynthesis up here being a change for the electron donor. But for a cyclic photosynthesis, that electron ends at chlorophyll, where the molecule began. The point is the final electron acceptor can vary as well as the electron donor. Any question about any of that? All right. That's it for chapter 6. Chapter 5, I mean. Any questions about chapter 5? Let me save that. And let's move on to chapter 6. right. I've got the initial slide giving you the major goals and a rough outline. Any question about any of this? Know the terms. We'll talk about each of them. 2, understand the various requirements for growth. 3, understand the 4 phases of growth. And 4, compare the different methods of measuring microbial growth. Any questions about this slide? Remember, when we're talking about microbial growth, we're talking about an increase in the number of cells, not, oh, come on, not an increase in the cell size. I used to teach middle school students, and obviously when we talked about their growing, we did not usually talk about reproduction.
Bacteria reproduce very simply and rapidly by doubling their contents and splitting in two. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
All right, any question about that? The requirements for all growth, including microbial growth, are twofold. They have to be physical requirements and chemical requirements. Some physical requirements include the temperature, the pH, the osmotic pressure, meaning the amount of salinity, etc. The chemical requirements, there must be a source of carbon for the cell, there must be a source of nitrogen, a source of sulfur, a source of phosphorus, a source of trace elements. If the cell requirements, there must be a source of oxygen. Obviously, if the cell is killed by oxygen, there must not be oxygen present, meaning an anaerobe needs to grow without oxygen. And there could be other growth factors which are required. If there are, like other organic growth factors required for the cells to grow, like Streptococcus pyogenes, the bacteria that causes strep throat, you need 2 organic growth factors that must be present or the cells will not grow. And we supply those organic growth factors by supplying red blood cells for the Streptococcus pyogenes to lyse the cells and then release the organic growth factors. I used to know what both of them were, but I only remember one of them. I think one of them is NADH. I don't remember what the second one is. It's something like factor V and factor something. Each species has its own minimum, maximum, and optimum growth temperature, meaning one species like Staphylococcus aureus has an optimum temperature that it'll grow as fast as that. It'll also have a maximum temperature which will grow, and if we go higher than that, the species cannot grow. And then a minimum temperature that it can grow at, and if we go colder than that, the species cannot grow. Any question about any of that? We can classify an organism into a group on the basis of their preferred temperature range. We have psychrophiles that grow around 10 degrees Celsius. That's the refrigeration temperatures, meaning the temperature of your refrigerator. There are psychotrophs. These are organisms that grow best around room temperature. There are mesophiles. These are organisms that grow best around 37 degrees Celsius. That's 98.6 degrees Fahrenheit. There are thermophiles, and they grow best around 60 to 62 degrees Celsius. And there's hyperthermophiles. They grow best around 95 degrees Celsius. Now, the temperature range of the various classifications are not rigidly defined. Like Streptococcus pyogenes is a human pathogen. It does grow best at 37 degrees Celsius, so we do call that a mesophile. But Micrococcus luteus that lives on the human skin doesn't actually grow best at 37 degrees Celsius because the human skin is cooler than 37 degrees Celsius. So it grows best, I think it grows best at 30 degrees, but maybe it's 35 degrees Celsius. A little bit cooler. And that's because the human skin is cooler than the internal body. And so Micrococcus luteus grows optimally a little bit lower than 37 degrees. We still call it a mesophile. So Micrococcus luteus is called a mesophile, and so is Staphylococcus. Well, that's not a good one. Streptococcus pyogenes, which grows best at 37 degrees. And not only can the mean vary a little bit, but the range can vary as well. So some mesophiles can grow best minimally at 10 degrees, others at high at 50 degrees, but others can grow all the way down to 0 degrees, maybe up higher than 50 degrees. And then some, like, I don't know, E. coli, probably can't grow at 10 degrees. So you need something like 20 degrees or at least 15 degrees before E. coli can grow. The point is this classification is not rigidly defined. The mesophiles are things that grow optimally around 37 degrees, but it can vary. Any question about any of that? All right, mesophiles include those things living in and on us, and all human pathogens are mesophiles. Mesophiles, most of them cannot grow in the refrigerator, 10 degrees Celsius, between 4 and 10 degrees Celsius, could be a little higher, depending on your refrigeration temperature. And that's because the cell membrane, as well as the proteins, will freeze in the refrigerator, and the protein will shrink in size, meaning become much more compacted than it normally should be. And so its three-dimensional shape will be changed, and so the protein cannot function. And the same thing with our mesophiles, and for as far as that goes, our cells and our enzymes. When you heat them up, they're going to start losing their three-dimensional shape and then totally become unwound. I don't know, here's a protein here. I don't know if you guys can see this. And it's three-dimensional shape. And you heat it up, the protein can unravel. Any questions about any of that? So somebody stated that when you have the protein denatured, the hydrogen bonds are breaking. That's probably true when you're heating things up. The hydrogen bonds are going to break, the protein will unravel. But in reality, you can form hydrogen bonds that shouldn't be there, as well as other bonds like ionic bonds. Like when you put the mesophile in the refrigerator, the protein, which is normally something like that, will crumple much closer, and then you get more hydrogen bonds forming. Any question about any of that? All right, let's talk a little bit more about temperature. At the wrong temperature, food can spoil. Let me blow this slide up so you can see it a little better. Bacteria can grow very rapidly between about 15 degrees Celsius and 50 to 52 degrees Celsius. And when the bacteria grow rapidly, they can spoil the food. Some bacteria can produce toxins. So you don't even need very much bacteria to grow before the toxins are produced. And so you want to keep the food out of this range for any period of time. So everybody knows this, once the meat is cooked, you should eat it, and you don't leave it out at room temperature. After about 4 hours, the meat should be put in the freezer or thrown out because it's been out at room temperature for too long. And this is room temperature, by the way, about 20 to 25, where bacteria can grow rapidly. So after about 4 hours, meat, once you've cooked it, it should be either put in the freezer or thrown out. I don't suggest putting it in the refrigerator because the bacteria has had 4 hours to grow, and the bacteria in the refrigerator could start spoiling the food, especially if you were to take it out of the refrigerator and then not eat it right away. So if it's been out for 4 hours or more, you should put it in the freezer or eat it immediately.
I have a question.
Uh-huh.
Like specifically about E. coli and like when meat gets contaminated, is it because the chickens eat something that is contaminated with E. coli? Like how do they get it? How does meat get infected by it?
Well, a lot of organisms have E. coli. Chicken, the problem is it has salmonella and one other human pathogen in its gut, and it lives in the chicken gut. So it's not a pathogen for the chicken, but for us, it's a pathogen because it doesn't live in our gut. And when you handle the chicken, because of the way they're clean, a lot of the chicken, at least 50% of them, have salmonella on the raw chicken. And it comes from just the cleaning of the chicken. It's the salmonella is inside the gut. When they clean it, they just reuse the water, and that's where the salmonella gets into the water, and then they wash the chicken, and so it gets on the chicken. And so raw chicken frequently has salmonella on it, why you should never obviously eat raw chicken or touch raw chicken and then not wash your hands and then eat.
So every chicken has salmonella.
Not every chicken, but most of them. So at least 50% of them, raw chicken.
How does that bacteria grow? Like where does it come from?
It's in the gut of the chicken.
It just
And
grows
then when
random.
they don't serve people the gut, so I don't know how they cut it out and clean it, but then they just shake the chicken in the water. And if you've ever seen the in process, they're pulling out the chicken and this water is kind of brownish or greenish looking. Okay, so my point is they reuse the water, and so salmonella frequently gets on the chicken. Does that make sense? So many raw chicken has salmonella on it. Not all of them, but I don't know, at least half of them. And and then, like I said, you touch raw chicken and then you don't wash your hands and you eat, you consume that salmonella. You touch the raw chicken and then you cook the chicken, and let's say you don't wash your hands and then you touch the cooked chicken, the salmonella from your hand get on the chicken.
Yeah,
Why
that makes sense.
never handle raw meat with something else that you're not cooking? Like making a salad. You should wash your hands before making the salad. Anyways, the point of this slide is that the bacteria can grow very rapidly in this temperature range. And it can grow in this temperature range between essentially 0 up to about 62 degrees Celsius. But the bacteria grow very slowly in this, I don't know, lighter pink color. There's no growth of bacteria below freezing. That would be not true if the bacteria can produce an antifreeze, but most don't. So the human pathogens certainly don't. And then there's no growth of bacteria above about 62 degrees. And that's not entirely true. The hypothermophiles can grow, but none of them cause human disease. And in fact, there are some Mexican women who put their corn on the cob in a gunny sack, and then they go to the volcanic stream, which is a hot water bubbling up from the volcanic vent, and they'll throw the corn in there. And there's lots of hypothermophile bacteria in that water. And then they cook the corn in the water, and then they take the corn home and feed it to their family, and nobody ever gets sick from it. So apparently the hypothermophiles don't tend to cause human illness, because obviously you're eating, they're probably dead or inactive hypothermophiles, because it's obviously not 95 degrees Celsius when you eat it, but it doesn't seem to cause human disease. No trouble in the people who do that. Any question about any of that? So psychotrophs and mesophiles can grow rapidly in this area and can cause food to spoil. And that's why if you have a large batch of food and then you put it in the refrigerator, the food will cool, but it doesn't drop through this temperature range quick enough. And it stays within this temperature range for several hours, giving the bacteria the time to produce a toxin like Bacillus cereus in a large batch of rice. If you have it in a large batch of rice and then you put it in the refrigerator, and this looks like it had about 3 hours at room temperature, and then they put it in the refrigerator, and that took 1, 2, 3, 4, 5, about 6 hours to go from this temperature, which that's way too hot for room temperature, but maybe the rice was still cooked and was cooling. And the point is you need to cool the food slowly. And what you can do with a large batch of rice is separate it into smaller batches, put the small batch in the refrigerator, and then the batch of rice will cool quicker in under 2 hours, going from above 40 degrees to below 15 degrees, in which case that's not enough time for Bacillus cereus or another bacteria to grow in the rice. Any question about any of that? All right. Another requirement of cells to grow is the pH. Most bacteria grow best around pH 7, so something like pH 6 to pH 8. And when you go below that pH 6 or above pH 8, the bacteria will stop growing or at least grow very slowly. This is actually a problem in a microbiology lab because in lab cultures, microbes tend to make acidic products from the media, and that acidic product can inhibit the growth. To minimize this slowdown of growth in the microbiology lab, we add a pH buffer to the culture media to allow the microbes to continue to grow in the media. Molds and yeast grow a little bit more acidic, meaning a little bit more lower pH than bacteria, and they grow best around pH 5 to pH 6. We do have, though, some bacteria that we call acidophiles, and these bacteria like to grow in acids, such as volcanic environments where the pH can range from 0 to pH 3. And there you actually know of one acetophile. What's a bacteria on or in you that is an acetophile? Come on, you just need to state the place where the bacteria grows. All right.
On your skin? Skin?
That is slightly acidic, but it's not this acidic. So that would be closer to a pH 5.
Oh, in your colon?
Not the colon, but in the stomach. The colon is actually alkaline. The stomach, though, is acidic. And the average pH of the stomach, at least when you're digesting food, is around pH 1.5. So the stomach is acidic. And we have acidophiles in our stomach, like Helicobacter pylori, a bacteria that we can find in your stomach. That's actually the bacteria that cause the stomach ulcers, by the way. Another requirement for growth is osmotic pressure. Microbes require a certain amount of salinity in order to grow. Most microbes that we grow in the lab require around isotonic solutions, meaning close to 0.9% sodium chloride. And if you go higher than 2% sodium chloride, most of the microbes will not grow. If you go less than that, I'm not sure how much less, but certainly close to 0%, most of the microbes will not grow. There are some microbes that actually like high osmotic pressures, such as in a hypertonic environment, where the solute concentration is higher outside the cell than inside the cell, like a bottle of molasses. If a bacteria, most bacteria like E. coli, falls into it, the water will leave the cell, and then we'll have plasmolysis of the E. coli, where water will leave the cell membrane will shrink away from the cell wall, and eventually that will kill the E. coli. Only takes a day maybe to kill the E. coli. E. coli is not very hardy. Staphylococcus aureus, though, would take longer, like maybe 2 weeks before you kill the Staphylococcus aureus. And that's why your bottle of molasses can survive at room temperature and not spoil, because of plasmolysis, preventing the growth of most bacteria, as well as most fungi.
And then you were saying last time that's the same reason why like honey lasts forever, right?
Yeah, honey and molasses, they can stay at room temperature essentially forever. My mother had a bottle of honey for 2 years. And if you remember, we had a bottle of molasses for 30 years. And my mother decided, you're not using this molasses. I'm not using it. I'm going to throw it away. But we figured it was at least 30 years old and it was still good. Although, to tell you the truth, we never checked it after 20 years. At 20 years, I made some molasses candy for my father, and he ate it and enjoyed it. But we never used it for 10 years. And so my mother decided finally to throw it away. High osmotic pressures can be used to preserve food, like salted vegetables, salted fish, salted meat. Dried fruit, you're having the sugar in the fruit creating a high osmotic pressure, and then that dry fruit can last, at least for several years. Any question about any of that? All right. So halophiles are organisms that like living in a high osmotic pressure, and halophile literally means lover, phile, halo, salt, so salt lover. There are two types of halophiles, an obligate halophile and a facultative halophile. An obligate halophile is something that requires a high salt concentration in order to grow. like some of the organisms, the microbes growing in the Dead Sea, that's between Jordan and Palestine, the West Bank. They're obligate halophiles. You take them out of that high salt environment, and that's close to 30% sodium chloride in the Dead Sea. The bacteria will die. So it requires that high salt concentration in order to grow. And then there are facultative halophiles. They do not require high salt concentrations to grow, but can tolerate much greater than 2% sodium chloride. Some can grow in 15% sodium chloride. Staphylococcus aureus is an facultative halophile. It can grow fine in a TSA plate, meaning TSA media, which is around isotonic, so about 0.9% sodium chloride. But it can also grow on your skin, which can get up to 20% sodium chloride. Obviously, it depends on how much you're sweating and when you last took a shower or a bath, but your skin can get up to 20% sodium chloride. And Staphylococcus aureus can grow on your skin. So it is a facultative halophile. Any question about any of that?
I had a question.
Huh?
So are there halophiles in the Great Salt Lake in Utah then?
I'm not sure of how much this sodium chloride is, but I'm sure it's at least the same as the ocean, in which
Mm-hmm.
case that would be above 2%.
Mm.
And so that would be, I don't know, at least slightly halophilic, but it's not a obligate halophile. That's a bad way to word it, but it's not something that's high salt. Like I said, the organisms growing in the Dead Sea, I know the Dead Sea is close to 30% sodium chloride. And above 30% sodium chloride, the salt will actually precipitate out of the water. And you can sometimes see in the Dead Sea salt crystals forming. And the Great Salt Lake in Utah, you'd have to tell me what the sodium chloride concentration is, because I just don't know off the top of my head. But yeah,
I'll look it up then.
it's at least somewhat halophilic, okay? Because I think it's more salty than ocean water. I used to know the salinity of ocean water, but I don't remember it. So, all right, you can also have growth in a hypotonic solution. This is a low osmotic pressure, like organisms growing in distilled water. Water can enter the cell by osmosis, and if the cell doesn't have a cell wall, the cell will lyse. As the water enters the cell membrane will expand. Then more water will enter the cell membrane will expand even further until the cell membrane pops, and that will kill the cell. If you ever put red blood cells in distilled water, within a minute, the red blood cells will all lyse and you'll stop seeing red in that water. Any questions about any of that? Obviously,
I looked it up and it
algae.
looks like it's the same as the ocean.
Okay, I didn't know that. I thought it'd be more salinic, salty. Okay. So it's a slightly halophilic environment.
Thank you.
All right. Like the algae growing in water that's not distilled water, which is essentially 0% sodium chloride, but it's very low, like the water in our streams. And plants or algae can grow in it, and they have a cell wall, and that prevents osmotic lysis. And then green photosynthetic bacteria can be in that water as well. And they'll have to have a cell wall. One chemical requirement is carbon. Typically, a cell has half of their dry weight due to the weight of carbon. So carbon is about half the dry weight of most cells. Carbon is used as a structural organic molecule and is an energy source for cells. Like for our cells, we get our carbon from glucose and other molecules in food. Heterotrophs use an organic carbon source for their source of carbon. An autotroph, though, can use an inorganic source of carbon. They can use CO2 dissolved in either the air or the water. Any questions about carbon and the chemical requirement? There are other chemical requirements. A cell has to have a nitrogen source, a sulfur source, and a phosphorus source. I'm not going to ask you the dry weight of nitrogen, but it is actually 14% of the dry weight of a cell. I will ask you where in our cells we have nitrogen, and the main source is in amino acids and proteins, but nitrogen can also be found in DNA, RNA, and ATP. Cells can often obtain their source of nitrogen from decomposition of proteins. Like that's where we get most of our nitrogen from, either from proteins or amino acids in our diet. Many bacteria also get their source of nitrogen from proteins or amino acids. So they just digest it to get their source of nitrogen. But many bacteria can use ammonium or ammonia as their nitrogen source. Some can use nitrate as their source of nitrogen. And if you don't know, green plants can use both ammonium and nitrate. They don't really like ammonium. If you give them too much ammonia or ammonium, that'll actually kill the green plant. But they love nitrate, and that's the main ingredient in a chemical fertilizer. A few bacteria can even get their nitrogen source from nitrogen gas in the atmosphere, and they can fix nitrogen, pulling nitrogen out of the air, and then bring it into the ecosystem. Any question about any of that? For sulfur, many organisms get their sulfur needs from proteins or amino acids. So sulfur is found in amino acids, at least some of them, like cysteine and methionine. You can also get sulfur from some B vitamins, and it's required for these B vitamins, thiamine and biotin. As well as sulfur is needed in DNA and RNA for the synthesis of DNA and RNA. Sulfur is usually obtained by either our cells or most bacteria from the decomposition of proteins. But there are many bacteria that can use sulfate for their sulfur source, and we cannot obviously use sulfate. Some bacteria can get their sulfur from hydrogen sulfide gas, H2S, and that's a toxin to us. Oops, didn't mean to do that. All right, all organisms also have a phosphorus requirement. Phosphorus is found in DNA, RNA, ATP, and then the membranes. Phospholipids are the main component of all membranes, including the cell membrane. Many organisms can get their phosphorus needs from phosphate. Even humans can get their phosphorus from phosphate. Any question about any of that? All right. All living cells do have trace elements that are required. Trace elements include inorganic elements required by microbes and other cells. in order to survive and maintain their cells and reproduce. And we call these inorganic elements trace elements. They're often required as enzyme cofactors. Some examples of trace elements include iron, copper, and zinc. Most organisms can get trace elements directly from their environment. And we get our trace elements from elements in the food we eat and the liquids we drink. Like if you get, I'm not sure where Vancouver gets its water from. Does the city of Vancouver get its water from the Bull Run Reservoir in Portland? Does anyone know? I'll have to look that up. Anyways, the city of Portland gets their water from the Bull Run Reservoir, and that water will have trace elements in the water. Well water will even have more trace elements than the Bull Run Reservoir. And so we can get trace elements in the water we drink. Because nobody,
The
almost
city of
nobody gets,
The city of Vancouver gets its water from the Orchards, Troutdale, and Sand and Gravel aquifers.
all right, so that's mostly wells. So then they will get trace elements in the well water. Like I said, there'll be more trace elements in the well water than from the Bull Run Reservoir, which gets its water from rainwater. And when it's rain, it's almost pure water. But obviously, the reservoir will put in a little bit of trace elements. And then you get trace elements too from the food you eat. And bacteria get their trace elements directly from their environment. Like the iron in the environment can diffuse into the bacteria. If an organism requires an organic growth factor, this organic growth factor is a requirement for cell growth. An organic growth factor is a biological molecule that the organism cannot synthesize itself. For humans, an example of an organic growth factor that we must get from our diet is vitamin C. Humans cannot make vitamin C. And all examples of vitamins, certain amino acids that we call essential amino acids, and certain essential fatty acids, we must get from our diet, and they are organic growth factors for us. Like the amino acid lysine. You must get that amino acid in your diet, which is pretty easy to get because if you just eat proteins, you're likely to get some lysine. But vitamin C, that's why we must eat generally fruit, especially citrus fruit, has vitamin C, as well as far as I know, all berries and many other fruits like watermelon. Even cabbage has vitamin C in it, and potatoes. In the old days, a major source of vitamin C was sauerkraut, because before refrigeration, people would ferment the cabbage to make sauerkraut, and then they would eat sauerkraut all winter long, and that would be their main source of vitamin C. Assuming they didn't grow in a place where they had fresh fruit in the winter. Some organisms also require purines and pyrimidines. And if they require it, that would be an organic growth factor. Any question about any of this? Molecular oxygen can be a requirement of cells to grow. If the cell requires oxygen, you must provide it or the cell cannot grow. But molecular oxygen is, in a sense, a toxic substance. All forms of oxygen can be made or converted into a toxic form of oxygen. And this usually happens in metabolism, such as in aerobic respiration. There are 4 toxic forms of oxygen. There's singlet oxygen, which is O2 gas, where the oxygen, electrons in oxygen are boosted to a high energy state. And then this oxygen is extremely reactive. Singlet oxygen can be found in phagocytic white blood cells. And then the phagocytic white blood cell will release the singlet oxygen on something that the phagocytic white blood cell wants to kill. So singlet oxygen is toxic. There's also superoxide free radicals. This is oxygen with an extra electron. That electron is unpaired, and so O2 with a -1 charge, and then that symbol there, let me blow that up so you see it better. That symbol there means a single oxygen atom, a single oxygen electron, sorry, giving this oxygen a -1 charge, but the one single extra electron doesn't like to remain this way. And then the oxygen either wants to become with another single electron, so O2 negative, meaning 2 negative, or it wants to give up that oxygen and become regular oxygen gas. And the point is, this is a free radical where the molecule has an extra electron that is unpaired. Free radicals are very unstable. They usually will bump into another molecule and then steal an electron from that other molecule. So the free radicals are highly unstable. Free radicals, like superoxide free radicals, are made in aerobic respiration. And then this superoxide molecule can steal an electron from DNA, or a molecule that bumps into DNA, which then steals it from the DNA, mutating the DNA. In our cells, we have an enzyme called superoxide dismutase, or SOD, which can combine the superoxide free radicals and hydrogen ions to make hydrogen peroxide and oxygen. But isn't hydrogen peroxide toxic to the cell? Yes, it is. But our cells have 2 enzymes, and our cells don't have 2 enzymes. Our cells have an enzyme to solve the hydrogen peroxide problem, meaning we have the enzyme catalase, which can convert hydrogen peroxide, shown here, into water and oxygen, shown here. And this catalase can work on any peroxide anion. This is a peroxide anion, oxygen 2, meaning oxygen gas with 2 extra electrons, which is toxic to cells, but we have the enzyme catalase to convert it into water and oxygen. Our cells don't have peroxidase, but some bacteria have the enzyme peroxidase, and they can take hydrogen peroxide and 2 hydrogen ions to make 2 water molecules. A bacteria that has peroxidase but not catalase can grow in the air, and Streptococcus pyogenes is as well as all members of the Streptococcus family, have peroxidase enzyme. And that's why Streptococcus can grow in the air, because they can convert the hydrogen peroxide to water. But Streptococcus family does not have catalase. Our cells do have catalase. And I just realized something here. This peroxide anion could come from this, when this superoxide free radical bumps into a molecule and steals an electron from it. That superoxide free radical will then be changed into a peroxide anion, which is also toxic to the cell. But we have catalase to solve it, and some bacteria have either catalase or peroxidase or both enzyme to solve the peroxide anion problem. Any question about any of that? Anyways, the peroxide anion, such as hydrogen peroxide, is the third toxic form of oxygen. The second toxic form being the superoxide free radical. And then the fourth toxic form of oxygen is a hydroxyl radical. This is the hydroxide anion losing one electron, so it has one unpaired electron. So this hydroxide ion has two extra electrons, meaning that they're paired, and there's a -1 charge, and this is fine in our cells. But this is not. This is a free radical, and whatever molecule it bumps into, it tends to steal an electron from it. Hydroxyl radical is actually made in aerobic respiration in trace amounts. It's also made by ionizing radiation. And whenever hydrogen peroxide reacts with certain metal ions, it makes hydroxyl radicals. Our cells have antioxidants in them, as well as bacteria, to neutralize the hydroxyl radical, as well as other free radicals. For example, our cells have vitamin C and vitamin E, which act as an antioxidant, and that will combine with the hydroxyl radical and then remove it. It does remove the vitamin C, but it's better for our cells to use up the vitamin C and then remove the hydroxyl radical from our cell. Any question about any of that? So vitamin C and vitamin E are antioxidants, which help remove hydroxyl radical as well as other free radicals from our cells. Any question about any of that? If not, I'm going to end it here, and I'll see you in the lab at 6:31. I'm a minute late, so I'll give you 1 minute. Any questions?
Nope.
I'll see you.